Hi viewers, welcome to SEKHAR'S Bio Factory for NEET Plant Kingdom with Pteridophyta general characters. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain the general characters of Pteridophyta. The term Pteridophyta, it was first time introduced by one of the scientists called Haeckel in 1866 year because these plants have feathery like leaves. The study of pteridophytes, especially their structure and life cycle, is called Pteridology. One of the scientists, Bauer, he was known as father of Pteridology, but Professor S.S. Beer, he was known as father of Indian Pteridology. One of the scientists, Edward Kelikowski, in the year of 1979, who can validly called as father of modern studies on pteridophytes genetics, and he published a summary and synthesis of the unique feature of homospores of pteridophytes. Pteridophytes are generally known as reptiles of plant kingdom or vascular amphibians or botanical snakes because these pteridophytes like reptiles, just like reptiles are the first land animals, similarly pteridophytes are the first land plants, so that pteridophytes are also called as reptiles of plant kingdom or botanical snakes. The smallest pteridophyte, it is Azolla, and along with that the largest pteridophytes like Cyanthea and Alsophia, these are called tree ferns. The evolutionary significance of pteridophytes is they were first vascular plants on land and first successful plants with true root, stem and leaves, but they do not bear fruits, flowers and seeds. They are earliestly known as vascular plants, which are originated in Silurian period 400 millions years ago in Palaeozoic Era and subsequently diversified and formed dominantly vegetation on Earth during Devonian to Permian period. These plants are found generally and grow on land which have soil surface and groundwater. The pteridophytes are generally present in cool, damp, and shady places, though some may flourish well in sandy soil condition. The dominant phase of plant body, it is a diploid sporophyte. These diploid sporophyte, it is well differentiated into a true stem, leaves, and roots. In pteridophytes, the roots are adventitious in nature and the stem is underground. It is called a rhizome. But some of the members may have aerial and erect stem like trees. In the structure of this plant body, the organ having well differentiated vascular tissues like xylem and phloem, so that pteridophytes are also called tracheophytes. But true vessels are absent in xylem, but except in some plants like Salacinella and Marsilia. In phloem, companion cells and sieve tubes are generally absent, whereas sieve cells only present in phloem. The secondary growth is unusual as they lack cambium cells and found exceptionally in Isoetes and Botrychium. Coming to Stelar System, the central vascular cylindrical axis in Pteridophytes, it is called stele. It is a primarily vascular tissue that includes pit, xylem, phloem, and pericycle. In it first appeared in one of the pteridophyte, it is called rhinia. Coming to types of steles, the first type is protostele. It is considered as the simplest type of stele without pit. In this haplostele, it is observed in Lygodium, actinostele observed in Silotum, Plectostele present in Lycopodium calvatum, mixed protostele, it is seen in one of the plant, it is the Lycopodium serenium. The second type of stele is ectophilic siphonostele. In this type, phloem is present on one side of the xylem only. Example, equisetum. The second type of siphonostele is amphibiolic siphonostele. In this, xylem is having phloem on its both sides. Such type of stele it is seen in Marsilea plant. The third type is selenostele. In this type, the stele with one or few non-overlapping leaf gaps. Such type of stele it is seen in Adiantum quadratum. The fourth type of stele is Dictyostele. In this, the stele with a large number of overlapping leaf gaps, it is seen in Dryopteris and Pteris plants. In Pteridophytes, the leaves are very long, large. Now such type of leaves are called as fronds. These frond leaves can be a pinnately compound leaf or simple type of leaf. These leaves have a dorsiventrally or radial symmetry with dichotomously branched with a lateral branched stem. The younger parts of the stem and younger leaves, a petiole and rachis of matured leaves contain a brown stiffy hair-like structures at bottom. These are called a ramenta. These ramenta it can protect you in function from mechanical injuries and desiccation. The young leaves are it shows a circinate venation. That means the young leaves are coiled like watch spring. This coiling brings growing point in center and it will be protected. There are leaf size variation among pteridophytes and they are basically two types. When leaves that are larger than stem and leaf gaps breaks the leaf traces, then such type of leaves are called as megaphyllous plants. The type of megaphyllous leaves are present in fern plants. When the leaves are shorter with a single leaf trace that is intact than plant, such type of leaves are called as microphyllous form. These microphyllous leaves are present in Salacinella. Usually, the leaves are photosynthetic in nature. Such leaves are called as trophophylous. But in pteridophytes, the leaf that can produce is sporangiums at ventral side. Now such leaves are called as sporophylls. The development of sporangium In pteridophytes, it may take place with the help of two methods. In these, the first method is leptosporangiate method. In this method, the development of sporangium takes place with the help of a single superficial cell. Now, this single cell can becomes into a complete sporangium. Such development is called leptosporangiate. Example, such type of sporangium development is seen in Pteris, Dryopteris and Adiantum plants. The second type of sporangium development is the sporangium development occurs from a group of superficial cells. Now this group of cells participate in the development of sporangium is called eusporangiate method. This eusporangiate method can be observed in some of the plants like Salacinella and Equisetum. The sporangia, these are present in group, is called sorus. Many sorus found on sporophyll. Now the group of this sorus is called sori. Inside this sporangium, some of the diploid spore mother cell, they can produce spores with the help of meiosis division. These are called meiospores. Now these spores are released to outside of the sporangium when its wall is break down. The formation of spores inside the sporangium, it maybe takes place with the help of two methods. In this, the first method is homosporous. Pteridophytes, if it can produces single type of or similar type of spores, now that method is called homosporous. Example, in Ferns, Adiantum, Pteris and Dryopteris, we can observe this homospore formation. Such type of similar type of spores can produces a gametophyte, it having both male and female reproductive organ. That means it can produces bisexual gametophyte. In heterosporous, only few pteridophytes can produces two different types of spores called megaspores and microspores. It's called heterosporous. Example, Salacinella, Marsilia, Salvinia, Azolla. These members can produce two types of spores in sporangium. These heterosporous can produce a gametophyte, but that is unisexual in nature, having either male or female reproductive organ separately. In some of the members like Salacinella and Equisetum, the sporophytes may also form a distinct compact structure. It is called strobili or cones. Ferns use both sexual and asexual reproduction methods for reproduction. Ferns can produce asexual. These are reproduced with the help of asexual method with the help of modified stem. It is called rhizome. Rhizome can spread just above or below the soil surface where they can form roots, and the undersides can produce new plants to apical or aerial portion. Some of the ferns have clumping forms and other have spreading habitat, but both kinds of reproduction takes place with the help of rhizome. In sexual reproduction, a haploid spore which is produced inside the sporangium, it can produces a haploid gametophyte. Now it is called prothallus. This prothallus is haploid in nature and it grows from a spore which is formed by meiosis division. This gametophyte of pteridophyte, it need cool, dry and shady places to grow. A gametophyte is the plant that can produces gametophyte. The fern gametophyte is small, bisexual and heart-shaped structure. Now it is called prothallus. This prothallus, it consists male and female reproductive organs for sexual reproduction. The sex organs present in the structure of prothallus, these are multicellular in nature, surrounded by jacket. The antheridium, it is the male reproductive organ, can produce flagellated male gametes. The archegonium is a female reproductive organ. It can produce a non-motile female gamete. The sperm or male gametes, these are flagellated in nature, which can swim and reach archegonium with the help of water. At this time, some of the chemicals like malic acid, it provides chemical stimulus. Male and female gametes are fused to form a diploid zygote. In pteridophytes, the formation of embryo in prothallus is called a diploid sporophyte. This development of embryo, takes place by two methods. One is called haploblastic method. In this method, the entire zygote itself, it becomes into embryo. Such type of development, it is seen in ferns. The second type is meroblastic development. When some parts of zygotes help in embryonic development, such type of embryo development, it is seen in Selaginella. The sporophyte, after its development, now it can becomes into a complete plant with a sporophytic plant body. In the life cycle of pteridophytes, alteration of generations observed in between heteromorphic and heterologous take place alternatively. These are the general characters of pteridophytes. Thank you for watching. If you like this video, subscribe my channel, share my videos to your students and friends.